Moving on to nerve impulse. Nerve impulse is a wave of electrochemical changes which travels along the length of the neuron, involving chemical reactions and movement of ions across the cell membrane. Electrical potential is a measure of the capacity to do electrical work. It represents a type of stored energy which is manifested during separation of charges across a barrier. In this case of neuron, the charges are positive and negative ions, and the charge separating barrier is the plasma membrane. The electrical potential that exists across the cell membrane is known as a membrane potential. A typical neuron at rest is more positive electrically outside than inside the cell membrane. This net difference is charge between the inner and outer surface of non-conducting neuron is called the resting membrane potential. The major factors which are involved in the resting membrane potential are number one, sodium potassium ions. One of many kinds of ions present in the nerve cells and the surrounding fluid. Sodium and potassium ions are the most important. Sodium ions are tenfold higher in concentration outside than inside the membrane surface, whereas potassium ions are 20 times more concentrated than the outside. All the neurons have very active sodium and potassium pumps located in their cell membranes, driven by the splitting of ATP. These pumps transport sodium out and potassium into the cell, both against their respective concentration gradients. For every 2 potassium that are actively transported inward, 3 sodium are pumped out. So inside becomes more negative than the outside of the cell membrane of neurons. Number 2, negative organic ions. The large negative organic ions such as protein, organic acids, etc. are much more inside the membrane than the outside, which they are only in negligible concentration. This makes the inside of neuron membrane more negative. Number 3, leakage of potassium ions from neurons. The cell membrane is virtually impermeable to all ions except potassium. Since the membrane is slightly permeable to potassium, some of it leaks out of the cell. The loss of this positive ion from the neuron by diffusion accounts for more negative charges inside than outside the cell membrane of neuron. No conduction of nerve impulse. As you can see over here is a diagram of resting potential. In the unstimulated state, a neuron has a membrane potential of approximately minus 70 millivolts. The relative concentration of the principal ions inside and outside the neuron are indicated by the sizes of the chemical symbols. Two of the major processes that contribute to the negative resting potential are the active exchange of potassium and sodium ions and the outward diffusion of potassium. The sodium potassium pump actively transports sodium out and potassium into the cell and is powered by the splitting of ATP by an associated enzyme ATPase. Initiation of nerve impulse. Under normal conditions, a nerve impulse is initiated by an appropriate stimulus called threshold stimulus applied at one end of the neuron, and it results in a remarkable localized change in the resting membrane potential. It disappears for a brief instant and is replaced by a new potential called action or active membrane potential, which is in the form of impulse. During this state, the inner membrane surface becomes more positive than the outside. This change is so brief, for perhaps a millisecond, that only a portion of the neuron is in the active membrane potential state. The major factors involved in changing the resting membrane potential to active membrane potential are 1. Sodium and potassium ion movement. The passage of nerve impulse is associated with increase in permeability of potassium ions. Sodium ions moving inwards, upsetting the potentially momentarily, making the inside more positive than outside. Neurophysiologists believe that the increased permeability is due to the opening of specific pores in the membrane, termed as the sodium gates. When these gates open, sodium ions rush into the neuron by diffusion, and some K positive ions move out. Number 2, charges are reversed. The inner side of the cell membrane has excess of positive ions and thus positive charges at its internal surface, and the outer surface becomes more negative. As you can see over here, the polarization, repolarization, hyperpolarization is shown in the graph as well as in the charges are also visualized. When a neuron is stimulated, the cell membrane at the point of stimulation undergoes a momentarily reversal in change dark color called an action potential. Perhaps for a millisecond, the inside of the membrane becomes positive relative to the outside. Sequence of membrane potential changes associated with an action potential. Resting potential, a polarized state. Sodium gets open and sodium diffuses into the cell, causing a depolarization of the membrane. Sodium gates close and potassium gates open. Potassium diffuses out, causing a repolarization of the membrane. Sodium-potassium pump restores original ion gradients and resting potential, which is the recovery stage, and step 2 to 5 take a mere 2 to 3 milliseconds. Number 3. Passage of nerve impulse. During active membrane potential, the neuron conducts the impulse in the form of nerve impulse. Number 4. Membrane potential. Active membrane potential of plus 0.05 volts plus 50 millivolts exists. These changes occur along the length of neuron till the impulse reaches synapse. Soon after passage of impulse, the resting membrane potential is restored by the movement of small number of ions, especially K positive moving out. This neuron now is ready to conduct another impulse. It may be added that in myelinated neurons, the impulse jumps from node to node of Ranvier. This is called the salted tree impulse. The normal speed of the nerve impulse in humans is 100 meters per second, but maximum speed recorded is 120 meters per second.